Assalamu alaikum wa rahmatullah. I am Assistant Lecturer Lamia Hadi. I will talk in this lecture's developmental of the internal and the external genital organs. Genital systems, introductions. The differentiated is a complex process that involves many genes, include some that are autosomal. The key to sexual dimorphism is the U chromosomes, which containing the testis determining gene is called a sex determining region on Y. In the short arm YP11, the sex determining region Y protein is the testis determining factors. Under it, influence male development occurs when absent female development is established. The male and the female reproductive system composed of the gonads, ovaries or testes, genital duct systems, mesonephric and paramesonephric duct, external genital. Initially, the gonads. The indifferent gonad. The sex of the embryo is determined at the time of the fertilizations. Gonads acquire male or female morphological in the 7th week of development. The morphological of the female and the male acquire in the 7th week of development. The gonad appear initially as a pair of longitudinal ridge. This ridge is called the genital ridge or gonadal ridge. Germ cells appear in the genital ridge in the 6th week of development. Germ cells appear in the genital ridge in the 6 weeks of development. The primordial germ cells are formed among the dorsal wall of the endodermal of the yolk sac, close to the allantois during the 3rd weeks. The germ cells formed in the 3rd weeks. These cells migrate by amoeboid movement along the dorsal mesentery of the hindgut to reach the gonads at the 5th weeks and invading the gonads in the 6th week. Reach gonads at the 5th week but invading the gonads in the 6th week. When the primordial germ cells, if the failure to reach the ridge, the gonad do not development. Shortly before and during the arrival of primordial germ cells, the epithelial cells of the genital ridge proliferate and the epithelial cells penetrating the underlying mesenchyma. They formed the primitive sex cords. Differentiated gonads. The male testis. If the embryo is genetically male, the primordial germ cells carry an XY sex chromosomes complex. The primitive sex cord proliferates further to forming to the testis cord or is called the medullary cord. Toward the hilum of the testis, the cord break up into the anestomosing of the tiny cells strand that later gives rise to tubules of the retitestis. During further development, a dense layers fibrous connective tissue that is called the tunica albuginea. Tunica albuginea separated the testis cord from the surface epithelium. You can see in this image the germ cells migrate in the along of the dorsal mesentery of the hindgut and forming to the genital ridge. Later, the epithelial or proliferative the primitive sex cord and forming to the testis cord or medullary cord. Toward the hilum of the testis, the cord will break up into the anestomosing of the tiny cells striated and later give rise to the retitestis cord. And dense fibrous connective tissue, tunica albuginea, separated the testis cord from the epithelial surface. In the fourth month, testis cord become horseshoe shaped and the end points are connecting with retitestis. A testis cord is now composed of the primitive germ cells and substanticular cells of the Sertoli cells. Sertoli cells derived from the surface epithelial of the genital ridge. The mesoderm of the ridge will give rise to the interstitial cells is called the Leydig cells or interstitial cells lying in the between cord and produce testosterone in the 8th weeks. It is cord at puberty forming seminephrous tubules. When seminephrous tubules are canalized, they join the retitestis tubules. You can see this is the seminephrous tubules canalized and join the retitestis tubules. Retitestis in turn enter the ductus efferens. With ductus efferens are remaining parts of the excretory tubules of the mesonephric systems. Ductus efferens link the retitestis and the mesonephric or Wolfen duct, which become the ducts deferens or vasodeferens. This is the efferent duct link between to the retitestis and in mesonephric or Wolfen duct, which become the ducts difference or البازل difference. The female ovaries. Female embryos with an XX sex chromosomes complement and no Y chromosomes. The primitive sex cord will be dissociated, forming irregular cells clusters. These clusters containing groups of the primitive germ cells and later then disappear and replaced by a vascular stroma forming the ovarian medulla. The surface epithelium of the female gonad, unlike that of the male, continues to proliferate. In the male, epithelial penetrating to the mesenchyme and forming to the primary cords, while in the female, this epithelial continues to proliferate. In the 7th week, this epithelial of the female genital ridge will proliferating, forming to the second cords. In the male, the second cord absent, while in the female, the epithelial of the genital ridge proliferates and forming to the second cords, or is called the cortical cords. These cortical cords near to the surface. In the 3rd month, these cords separated also into the cell clusters, forming to the ovarian follicles or follicle cells. These follicles surrounded each oogonia. Oogonia derived from the germ cells. Okay? Now, a difference between to the male and the female. The surface epithelial of the male proliferating and penetrate to the epithelial of the mesenchyme, forming to the primitive cords, while in the female, this epithelial continues to proliferate, and in the 7th week, this epithelial forming to the second cord is called to the cortical cords. In the 3rd month, these cords separated in the cell clusters and forming to the follicle cells of the ovarian cortical, and surrounded each oogonia, and oogonia derived from the germ cells. Together, the oogonia and the follicle cells constitute the primary follicle cells. Okay, each follicle surrounded the oogonia, and the follicle cells constitute the primary follicle cells. Now, the diagram show the influence primordial germ cells on the indifferent gonad from the male and the female. Now in the male, containing to the 44 somatic cells plus XY, while in the female 44 plus XX. المale الواي influence الindifferent الgonad forming to the testes, while in the female absent of the Y, the indifferent gonad forming to the ovary. The testes forming to the medullary cord development from the primitive cord proliferate and forming to the medullary cord, while in the female the medullary cord degenerate from the cluster cells and these cells degenerate forming to the stroma, later forming to the vesicular of the medulla, ovarian medulla. In the male, no cortical cords, primitive cells or cords proliferate forming to the medullary cord, while in the female the primitive cords proliferate and forming to the secondary cords, or is called the cortical cords. In the testes have a thick tunica albuginea, while in the female no tunica albuginea. Now genital duct. First, in different stages. Initially, both the male and the female embryos have two pairs of duct lateral to the genital ridge. The first, mesonephric duct, or is called Wolfen duct. Wolfen duct plays an important part in the development of the male reproductive systems. Wolfen duct, important part of the development of the male reproductive systems. Second, paramesonephric or Mullerian duct. Mullerian duct have a leading role in the development of the female reproductive systems. إذن Mullerian duct هو يكون important in the development of the female reproductive systems, while Wolfen duct important part in the development of the male reproductive systems. Mesonephric duct arise as invaginations of the epithelial surface of the urogenital ridge. Cranially, the paramesonephric duct opening into the abdominal cavity with funnel-like structures. Caudally, the paramesonephric duct first run lateral to the mesonephric duct, then crosses its ventral to be middle to it. In the midline, the right and the left paramesonephric duct fuse to forming the uterine canal that project caudally into the wall of the urogenital sinuses. producing the paramesonephric or Mullerian tubercles. You can see Uterine produced to the paramesonephric tubercle or Mullerian tubercle. Mesonephric duct opening into the urogenital sinuses on the side of the Mullerian tubercle. Here you can see the paramesonephric duct lateral to the Wolfen duct, and in the middle, right and left of the Mullerian duct fuse forming to the uterine canals that project caudally into the wall of the urogenital sinuses and produce the Mullerian tubercle. Right and left Mullerian duct fused forming to the uterine canal. Now differentiated genital duct. First, male genital duct. Caudal excretory tubules of the mesonephros will not disappear, forming the efferent ductuli from the epigenital tubules. The efferent ductuli connecting the retitestis and the vas deferens, or is called ductus efferens. Also, these tubules will forming a vestigial of the paradidymis from the paragenital tubules. The cranial part of the mesonephric duct degenerates, forming the vestigial of the appendix, epididymis. Caudal excretory tubules forming efferent ductuli, connecting the retitestis and vasa deferens. Also, these tubules forming a vestigial of the paradidymis from the paragenital tubules, while the cranial part of the mesonephric duct degenerations and forming a vestigial of the appendix epididymis. Remaining part of the mesonephric duct at their cranial part form the duct of the epididymis is the ductus deferens, or is called the vas deferens, and ejaculatory duct. Seminal vesicles outbud from the mesonephric duct at the distal end of the vas deferens, and the paramesonephric duct degenerate except for the small portions at their cranial end, forming the appendix testis. Paramesonephric duct develop into the main genital duct in the female, initially, which have three parts from cranial to caudal. Cranial ventricle portions that opening into the abdomen cavity. Horizontal part that cross the mesonephric duct. Caudal ventricle part that fused with pattern from the opposite sides. Cranial and horizontal part development into the uterine tubes. The caudal part fused to form the uterus. After the duct fused in the midline, the board ligament of the uterus is established. The fused paramesonephric duct give rise to the corpus and cervix of the uterus. They are surrounded by layers of mesenchyme that form a muscular coat of the uterus. The myometrium and its peritoneal covering the perimetrium. Myometrium and the perimyometrium and the peritoneal covering. At the point of the paramesonephric tubercle that they produce from the uterine canal, you can see two sino-vaginal bulbs grow from the pelvic part of the urogenital sinuses and forming to the vaginal plate. This plate is increased in size and canalized in the 5th month, forming the vagina. The vagina has a dual organs or double organs. The upper part of the vagina near the uterus that called the pharynx. Pharynx derived from the uterine canal. Uterine canals form from the fused from the two parts of the paramesonephric duct, left and right, forming to the uterine canals. The lower part of the vagina derived from the urogenital sinuses. The vagina has double organs, the upper part from the uterine canal from the fused of the right and the left of the paramesonephric duct. The lower part of the vagina derived from the urogenital sinuses. The pelvic part of the urogenital cavity is separated from the vaginal cavity by a thin tissue plate is called the hymen, which is derived from the epithelial of both these structures. The hymen, thin tissue plate derived from the epithelial of the both structures. A remnant of the cranial and the caudal mesonephric excretory tubules may form the vestigial of the apophorin and the paraphorin respectively inside the mesovasaria. And remnant of the cranial mesonephric ducts seen in the apophorin and of the caudal mesonephric duct seen the wall of the vagina or the uterus as corner cysts. Now, the clinical correlates uterine and vaginal defects. First, duplications of the uterus. It may be result from the abnormality in the effusions of the paramesonephric ducts at the uterine canal. These anomalies may be of in many types. May be complete. Is called the uterus didelphysed with a double vagina, or an indent uterus, or is called the uterus arcutus مقوس يصير أو مسنن. Two uterine horn and one vagina is called the uterus bicornus, or maybe atresia of the paramesonephric duct resulting in uterus bicorns with rudimentary horn, or atresia of the uterine cervix. Or abnormal sino-vaginal bulbus that may be result in the double vagina or atresia of the vagina. Now, development of the external genitalia. First, in different stages. The external genitalia develops during 3 to 6 weeks of the gestations. The cloaca membrane is surrounded by a bilateral mesodermal cloaca fold. These folds fused anteriorly, forming the genital tubercle. Caudally, these folds are subdivided in two parts. The urethral fold anteriorly that surrounded a urogenital groove. Second, the anal fold posteriorly. Okay. At the same time, the urethral fold becomes surrounded by the bilateral genital swelling that are the primordial of the scrotum of the male and the labia major of the female. Now, the differentiated external genitalia. First, of the male, the genitalia tubercle, you will elongate it, forming الفلس. الفلس is the primordial of the penis and glans penis. The urethral fold elongated, forming the urogenital groove or the urethral grooves that lies on the caudal side of the phallus. These grooves, urethral grooves, is lined by the endodermal of the urethral plate. The urethral fold will be closed to form penile urethral at the end of the third. This part of the urethra never reached to the tip of the phallus, therefore the ectoderm at the tip of the phallus is invaginated to form a cord tissue between phallus and penile urethra. Canalized of this cord will form the external urethral mucus. All these development changing occur by the effect of the androgen testosterone hormones secreted by the testes. Now the clinical correlates. First, the hypospadias occur by a failure of the closure of the urethral fold, also is called scrotal fold. It causes abnormal urethral opening at the inferior aspect of the penis. Massive failure will result in the separations of the scrotal swelling. They may simulate the labia majora. The second, epispadias, occur if the genital tubercle is formed at the caudal end of the cloaca membrane, normal in the cranial positions. It associated with the abnormal urethral opening at the dorsum of the penis. Extrophy of the bladder, failure of the mesoderm, migrations at the primitive streak toward the cloaca membrane. It may be associated with epispadias. 4. Micro penis. 5. Pift penis or double penis. Now the external genital of the female. The short genital tubercle will forming the clitoris, the infused urethral fold forming labia minora, urogenital groove form the vestibule of the vagina. Ultrasonic examinations of the fetus during the third and the fourth month may lead to wrong diagnosis of the male sex, because in this stage the female has a larger genital tubercle. Now the clinical correlate a defect in the schedule differentiated of the external genitalia could be seen in the first Klinefelter syndrome with karyotype 47 XXY or other variants, example XXY. A second, gonadal dysgenesis, abnormality in the sex region on Y gene. Third, pseudo-hermaphroditus occurring in the male or in the female. 4. Testicular feminizations occurring due to insensitively to androgen. Now, descending of the testes, the gonads are attached to the posterior abdominal wall by the urogenital mesentery. The caudal part of this mesentery is called the caudal genital ligament. The gilbernaculum is the mesenchymal condensations extending between the caudal poles of the testes to the scrotal swelling outside the abdominal cavity. This gilbernaculum grows outside the abdomen and thus pulling the testes down. This pulling is assisted by increased intra-abdominal pressures that occurs by the growth of the abdominal viscera. Later on, the extra abdominal part of the gilbernaculum regressive and thus completing the passage of the testes throughout the inguinal canal down toward the scrotum. The peritoneal evagination surrounded the descending testes and forming the processes vaginalis and tunica vaginalis. The descending testes are also covered by internal spermatic fascia, the cremasteric fascia, and the external spermatic fascia. These three fascial layers are derived from the muscles of the interiorly abdominal wall. The above developmental changing occur under the effect of the androgen hormone. Now, the descending of the ovary. This descending occurs in the less degree than the descending of the testes. The ovary reaches just below the pelvic brim. The cranial part of the urogenital mesentery forming a genital ligament. The cranial part of the urogenital mesentery forming a genital ligament. A genital ligament that development and forming the suspensory ligament of the ovary. Then, a suspensory ligament derived from the genital ligament in the cranial part of the urogenital mesentery, while the caudal part of the mesentery forming a caudal genital ligament, with caudal genital ligament forming a ligament of the ovary and a rounded ligament of the uterus. Thank you.